Hey everyone, and welcome to Angiospermss with Ashley. Okay, so today we are going to be continuing going through our PowerPoint on the seed plants, and we'll be finishing up this with our angiosperms. So we'll go ahead and get started. Pull this up. Okay, so to start off, we're talking about angiosperms. This is going to be our most diverse group of land plants and often one of the favorites of our students because they are often the most beautiful as well. This is due to them having flowers for reproductive purposes as well as seeds and fruit. The ovule is going to develop into the ovary of our flower, which is unique to angiosperms. Also unique to angiosperms is that we see double fertilization occurring, which means two sperms are fertilizing our megagametophyte or our female gametophyte, and we'll get more into that in a few minutes. They have also evolved complex reproductive strategies and co-evolved with insects. So this is really where we see all of this variation in the color and shapes of our petals of flowers. This is generally a reproductive strategy to get pollinators to take their pollen with them. And they also have a lot of symbiotic relationships with many different kinds of insects to increase fertilization. Some of the older lineages are going to include Ambarella, Nymphalis, which is our water lilies, Austrobaileos, which is our star anise, and then the Magnolids, which is our magnolias. And then the two main groups where we see a lot of our diversity is going to be in the monocots and the eudicots. So looking at our angiosperm phylogeny, Ambarella is considered a sister lineage to all the other flowering plants. So here we have those Nymphalis and our Austrobaileos that were also our ancestral groups. And our magnolias, monocots, and eudicots are all pretty closely related here. They were able to figure this out using molecular and phylogenetic analyses. And these are kind of supposed to be our ancestral flowering plants, and they're pretty cool looking, I think. So you may remember us talking about in gymnosperms, the woody xylem tissue being primarily or really only tracheids. But in angiosperms, we are going to see that the xylem tissue will consist of both tracheids and vessel elements. So tracheids are generally going to be much smaller in diameter and have those tapered ends that we mentioned last time. Our vessel elements, this is a vessel element here, are generally much wider and just big. So they are going to be much more efficient at water transport through the cells. Even though they're both still dead, these just have much more volume to transport water. And vessel elements also have perforation plates along the ends of them that will allow for better water transport, apart from just having these pits that tracheids have. So looking a little more at the vessel element anatomy, here are those perforation plates that I mentioned. So this is going to allow water to kind of move more in a vertical way rather than having to just diffuse through each of the tracheid cells. That's kind of it with these. Looking at the phloem tissue, we're going to see kind of similar to our vessel elements, we'll have these sieve plates that are kind of like those perforation plates that are going to assist in just transport of sugars and other metabolites through our sieves. And it also acts as a protective mechanism against any injury. So if the phloem tube were to somehow become damaged, or if one of the companion cells that I'll talk about in a second were to die, and then therefore the phloem cell would die, because remember our phloem cells are living, then these sieve plates can serve to block the flow of any more phloem sap so that you don't lose any of the metabolites that have been obtained whenever there is an injury. So those companion cells that I briefly mentioned before serve to really be a companion to our sieve cells. These sieve cells don't have all of the essential organelles to sustain life on their own. So the companion cells do contain those and are able to help metabolically maintain and sustain these cells, which is pretty cool. So if the companion cell dies, so does our sieve cell. Comparing our gymnosperm and angiosperm phloem, we see that the gymnosperms, they do have sieve cells, and they just have sieve areas. So these are kind of those pores that allow sugars to be transported throughout. But in our angiosperms, we have these sieve tubes, as well as we have these sieve plates, and we have sieve areas, and the companion cells. So the pores here are generally much larger, and these sieve tubes are much more efficient at sugar transport than just what we've got in our gymnosperms. So angiosperms did evolve after our gymnosperms, and we can see evidence of this even in just our xylem and phloem at how much more efficient they are at transport. And here's an example of gymnosperm and angiosperm wood. If you remember from the last lecture, I mentioned that gymnosperms typically are considered softwood and angiosperms are hardwood. You can even see this. We generally have a much more intense ring pattern here in our angiosperm tree, which is, this is going to be red oak, that's an angiosperm, and red pine is a gymnosperm. So we see the cells are generally much larger here in our oak, and this is because of that vascular bundle that is our xylem and phloem. Those are much bigger cells typically, so then we see more of a pattern like this rather than in the smaller case of our gymnosperms. So moving right along into the flowers, there typically are going to be 4 whorls of a flower starting here at our sepals. So the sepals are going to consist of leaf organelles or organs that are going to initially serve to protect the flower bud as it's still developing. And then whenever the flower is ready to burst, then the sepals will open up and create this structure here. Our next whorl is going to consist of our petals, and altogether these are considered our corolla. I don't think I mentioned this. Altogether, the sepals are our calyx. And then all of our petals are our corolla. And these are just going to be modified leaves that primarily serve to attract our pollinators. Our third whorl is going to consist of our stamens, which will be the male parts, which is the filament that holds our anther, which is going to produce our pollen grains. And then lastly, in our fourth whorl, we have primarily our female parts. So the whole thing is considered our carpel, and this is going to be our stigma, style, and ovary. And the ovary is going to contain our ovule, which will then eventually produce our megaspore. And the pollen tube, as we'll get into in just a moment, will develop when the pollen lands on top of our stigma. The pollen tube will move down through our style into our ovary and hopefully find an ovule. So a little close up here of our anthers and stamen. The anther, if you were to cut it in half and look inside, you would find 4 pollen sacs, and here's a cross section of that to see it in real life. So if you were to cut that open, you'd see these 4 pollen sacs, and each of them will contain a bunch of pollen grains that will be released when the plant is ready. So we're going to kind of focus in, as we're about to get into double fertilization, on the carpel area. So we have our stigma, our style, and our ovary. If you look at the cross section of this flower, you can see that even better with our stigma, style, and ovary. Our ovule sits within that, and typically there's a bunch of ovules inside, but for the sake of simplicity, we're just going to look at one. So now here we are getting into this double fertilization. So here is our stigma, or our carpel, sorry, that is going to consist of our stigma, style, and ovary. The pollen grain, or our male gametophyte, will land on top of our stigma. And then we'll start to develop a pollen tube that will move down the style into our ovary and find an ovule. Once it has found an ovule, 2 sperm cells will be moved down the pollen tube to our ovule, and then we are here at our double fertilization. So inside the ovule, the megasporangium, which is diploid, will form a megaspore mother cell, which will then produce 4 haploid megaspores through meiosis. So then our megaspores are haploid. Only one megaspore will survive, and this megaspore through mitosis will develop into our megagametophyte, which is the 7-celled embryo sac. So looking at that a little further, within this 7-celled embryo sac, we have our egg, which is going to sit really close to where the pollen tube has entered through the micropyle, which is just an opening in the ovule. The egg will be situated there with the help of the synergids. The synergids are 2 cells that will help direct the pollen tube growth and make sure that the sperm are able to reach our egg. We also have our polar nuclei, which is going to have 2 nuclei, which I'll get into in just a second. And then we have 3 antipodal cells. So what happens when we have these 2 sperm cells come down our pollen tube and enter through the micropyle, one of these sperm cells will proceed to fertilize our egg, producing our diploid zygote that will grow into our next sporophyte generation. And one sperm cell will bypass this and find our polar nuclei, which I remind you has 2 nuclei already. This sperm will then fertilize this polar nuclei, creating a triploid endosperm. This triploid endosperm that forms when it's fertilized will become the food source for this growing embryo that was just formed. These 3 antipodal cells will also serve to increase the nutritional content of the endosperm for our growing zygote. So now we are going to look at the life cycle of our flower, now that we know a little bit about our double fertilization. So we're going to start out with our mature sporophyte, which is diploid. Looking at the male side of things, when the anther is ready through meiosis, it will develop microspores in its microsporangium. These microspores are produced through meiosis, so this is going to be haploid, and this is what will form our pollen grain. On the female side of things, within our ovary and ovule, the megasporangium, which is diploid, will produce our megaspore mother cell, and then that will produce, through meiosis, 4 haploid megaspores. Only one of these megaspores will survive, and that will go on to create our megagametophyte through mitosis. So here we have our female gametophyte or our megagametophyte, and it is haploid at this point. At the same time, these microspores that are haploid, through mitosis, will begin to produce our pollen grains, which are our male gametophytes. These pollen grains will be dispersed through either wind or animals or whatever means it can, maybe our cars in the spring, and will hopefully eventually land on top of our stigma, so it can produce that pollen tube and find the ovule where the egg is being held. At this point, that double fertilization we just went over will occur. So the egg will become fertilized, creating our diploid zygote. And at the same time, the endosperm, the triploid endosperm, will form that nutritive tissue for that zygote to survive from. Through mitosis, a fruit will be developed that will contain the seed, which will also contain our growing diploid embryo. The seed will be dispersed through wind or animals or water. And eventually, once it lands in the right place, in the right environment, it will begin developing into our sporophyte. And once this sporophyte has matured, the process will begin again. So I had mentioned when we first started our presentation that the 2 most common groups to find our angiosperms in are our monocots and our dicots, or our eudicots. So I want to look at a few differences between these just to outline those. So in our monocots, first looking at our cotyledons. So the cotyledon is going to be the first leaf that will be produced from the plant. In monocots, we only get 1 cotyledon. So here you can see that cotyledon being developed. And in dicots or eudicots, we will see 2 cotyledons. So here we have 2 first leaves that will erupt once the zygote has, or our sporophyte has begun to grow. Looking at our vascular tissue in monocots, the vascular tissue, which is our xylem and phloem, producing our vascular bundles that are these purple spots, they are just scattered throughout the whole stem. So that also leads us to see these parallel veins or this parallel venation within our leaves. In dicots, we see the vascular bundles are actually arranged in a circular arrangement around our stem. And this helps contribute to these branching veins that we see in our leaves. And lastly, looking at our flowers, monocots have petals in multiples of 3, and dicots have petals in multiples of 4 or 5. And here we actually see that these lower 3 petals are actually our sepals in this case, but either way, we're seeing 3 or 6 petals, whichever way you like to look at it. And here we're seeing 5. Okay, so we have just finished talking about angiosperms. Definitely recommend going back over and reviewing all of that with your handout and just going back over the life cycle and the double fertilization again, maybe talking about it out loud. And then you can go ahead and complete the rest of the activities for exercise 4, our seed plants. So thanks, y'all.